Hi everyone, welcome to my channel Microbiology with Sumi. If you like my video, please like, share and subscribe to my channel. Today's topic is multiplication of viruses. So let's start with viruses. Viruses are obligate intracellular parasite. They depend on host cell for its multiplication. So viruses are parasites which penetrate inside the cell and carry out its multiplication inside the host cell. Further, viruses multiply and carry out cell division in a complex way inside the host cell. Viruses go through 6 steps of multiplication. In multiplication of viruses, the first step is adsorption or attachment, that is attachment of a virus cell to the cell surface. Second is penetration, the penetration of entire virus or the genome of virus. Third is uncoating, that is removal of the external parts, that is capsid or envelope of virus, and then penetration of the viral genome inside the cell. Fourth is biosynthesis, synthesis of various molecules that are required for formation of virus. Assembly and maturation, after synthesis of various biomolecules required for multiplication of virus, these biomolecules assemble and carry out its maturation, and further they release out of the host cell. Now let's see this multiplication steps in detail. The first one is adsorption or attachment. Viruses and host cell come in contact with each other by collision. Now in nature, host cell and viruses collide with each other. Now if the virus and host cell have affinity towards each other, then the virus gets attached to the host cell. So affinity between host and virus cell is important for attachment. The host cell should contain a specific receptor site. To this specific receptor site, virus gets attached and carry out its adsorption step. So for carrying out the adsorption step, it is important that host and virus cell, first of all, they collide to each other, and if they have affinity towards each other, then the virus cell gets attached to host surface, and before attaching to host surface, the host surface should contain a specific receptor site. Then only the virus cell can attach on host surface on this receptor site and adsorption procedure can be carried out. The second step is penetration. After attachment of virus to the surface of cell, it carries out penetration. In case of bacterial cell, only nucleic acid is introduced in the cell by a complex mechanism. As the cell wall of bacteria is very complex, only nucleic acid gets penetrated inside the cell. Now in case of bacterial cell, the cell wall of bacterial cell is very complex in nature. So entire virus cannot enter in the bacterial cell. Only the nucleic acid is introduced in the cell by a complex mechanism. Whereas cell wall of animal cell is not as rigid as cell wall of bacterial cell. It is quite flexible. So entire virus can enter inside the cell. Now in case of animal cell, its cell wall is flexible. So entire virus can enter in the cell easily. Further, virus can enter inside animal cell by a mechanism called as viropexis, which is similar to phagocytosis. In this mechanism, virus fuses to plasma membrane and release viral genome in cytoplasm. Now, in the mechanism of penetration regarding animal cell, this is carried out by a procedure called as viropexis, and this procedure is similar to phagocytosis. Now in this mechanism, virus cell fuses to the plasma membrane of animal cell and releases its genome in the cytoplasm. Now this was all about penetration. Third step is uncoating. Now, uncoating is a procedure of removal of outermost membrane, capsid and release of nucleic acid in the cell. Now, as we all know, and some viruses contain envelope, but all of the viruses contain the capsid. So here outer membrane is considered as capsid. Now in uncoating procedure, the removal of this capsid is done and then the nucleic acid is released inside the cell. Now, lysosomal enzymes of host cell helps in uncoating of virus. In this procedure of uncoating, the lysosomal enzymes remove the outer coat of virus in a phagocytic vacuole. As we all know, it carries out by a viropexis mechanism which is similar to phagocytosis. So here in this procedure, lysosomal enzyme removes the outer coat of virus in phagocytic vacuole and the internal proteins and phagocytic genome is released in the cytoplasm of cell. Further, the viral enzyme uncoats the virus and release the genome. So here the viral enzyme which is present, it uncoats the virus. So uncoats means it removes the capsid and then release its genome. The fourth step is biosynthesis. In the step of biosynthesis, the essential biological elements which are required for synthesis of virus are formed. So whatever molecules are required for formation of virus are synthesized in this step. Here nucleic acid, capsid protein and various enzymes are synthesized. Generally, DNA virus synthesize the DNA in the host nucleus, but in case of RNA virus, all components are synthesized in cytoplasm. So, in case of DNA viruses, the synthesis of DNA virus is carried out in the nucleus of the host cell. But in case of RNA virus, all components are synthesized in the cytoplasm. They are not synthesized in the nucleus. In biosynthesis mechanism, the viral nucleic acid is transcribed in mRNA molecule. Now, when a biosynthesis mechanism is carried out, the nucleic acid of virus, first of all, it is transcribed in mRNA molecule. Further, this mRNA molecule undergo translation process to form non-structural proteins. So, in replication of viruses, the first step is transcription and second is translation. So here also first the nucleic acid is transcribed to mRNA and then the mRNA is translated to various non-structural protein molecules. Third one is the non-structural protein further starts synthesis of viral components. Now this proteins, non-structural proteins are the base that are used for synthesis of different components of viruses. And further viral nucleic acid is replicated. The fifth step is assembly or maturation. Viral nucleic acid and protein comes together to form a nucleic capsid. Now when all components are synthesized, the nucleic acid and the protein, they come together and form the important structure of virus, that is nucleocapsid. DNA virus assemble in nucleic acid of host as I told you before, and RNA virus assemble in cytoplasm of host. The last step is release. The progeny of virus is released by lysis of host cell. Release of animal virus occurs by process of budding without lysis of cell. Now normally when the virus cell releases, it causes lysis of host cell, but in case of animal viruses, these virus progeny is released by a procedure called as budding, and here lysis of cell is not done. In case of envelope virus, they take a part of cell membrane and form an envelope around themselves. And after release of virus, they infect the neighboring cell. Now whenever the virus is formed, it bursts or it carry out lysis of cell, or they get out from the cell by the procedure of budding. They infect the cells that are present in the surrounding and they spread in such a way. Now what is eclipse phase? The eclipse phase is a time interval from penetration of virus in the host cell till appearance of viral progeny. So all the time that is required from entrance, that is penetration of viral genome or viral cell in the host cell till the formation of virus progeny is called as eclipse phase. Thank you for watching my video. If you like my video, please like, share and subscribe to my channel, and don't forget to comment in the comment section.